Good morning to one and all. I am Dr. E. Susitra, working as Assistant Professor in Department of Pharmacognosy at School of Pharmaceutical Sciences, Wales Institute of Science, Technology and Advanced Studies, Chennai. My topic of presentation is Introduction to Pharmacognosy with a brief note to the definition, history, scope and development of pharmacognosy. The learning outcomes will be, at the end of the presentation, the learners will be able to know the definition of pharmacognosy and some common terms which is related to pharmacognosy and phytopharmaceuticals, a brief note about the history of pharmacognosy both at ancient times and modern times, and also the learners will be able to understand the scope of pharmacognosy in different fields and the modern concepts which are involved in development of pharmacognosy. So we are all aware of the term botany. What does this pharmacognosy mean? Pharmacognosy is derived from two Latin terms. Pharmakon means a drug and gignoso means to acquire knowledge of. So pharmacognosy is a branch of science which deals with the study of crude drugs obtained from a natural source, which can either be a plant source, animal source, mineral kingdom or even microorganisms. The study of these crude drugs obtained from the natural source and their constituents is called as pharmacognosy. Initially the term pharmacognosy was known by the term materia medica. Mostly, the crude drugs used in medicines often come from plant source, and a very few number has been reported from animal and mineral kingdom. A few plant sources include entire plants, leaves, pods, or any part of the plant like seed, root, bark, stem, rhizome, etc. Sometimes, in fresh conditions, certain parts of the plant are used like peels and corms, but most of the drugs are dried after collecting it. Not only these, the crude drugs are also obtained in an indirect method. To mention a few, cotton, silk, jute, nylon, which plays a specific role in surgical dressings, and also the minerals like kaolin, diatomite, which finds use as a pharmaceutical aid, and as well, the dried juices obtained from the leaves, the latex obtained from the capsules, the dried aqueous extracts obtained from the woody region, and some exudates like gums, volatile oils, fixed oils, resins, balsam, etc. all come under the classification of crude drugs. The animal sources include an entire animal part as seen in cantharites, or it can be a glandular product like thyroid organ, or it can be an extract such as a liver extract taken from an animal tissue, or it can be a product like fish liver oil, musk or beeswax hormones, enzymes and antitoxins, etc. Broadly speaking, the term pharmacognosy has been classified as organized and unorganized. The difference between the two being, organized are the ones which are taken from the direct part of the plants where you can give a cellular organization or a pattern, whereas the unorganized drugs are nothing but those that are not obtained from the direct part of the plant and which can be prepared by a simple physical process like incision, means cutting, or it can be either drying or extracting procedures. So these does not have any cellular structures as we have seen in the case of certain crude drugs like gums, waxes, liquid extracts, leafy juices, etc. Let us see the ancient civilization. Broadly speaking, the history of pharmacognosy dates back to the Western medicine, which includes the Mesopotamians and the Egyptians, and next the Unani system of medicine, which dates back to 980 to 1037 AD, and our Indian system of medicine, namely the Rig Veda, Atharva Veda, and the most important one, Ayurveda, dating to 2500 to 600 BC, the Orient herbalism, which includes the Chinese, Japan, and Tibetan medicinal plants, and last but not the least, the Greek and the Roman herbalism. So this ancient Egypt, mostly the written documents are given in the form of paper, mentioned as Ebers Papyrus in the period 1550 BC, where around 800 prescriptions containing 700 drugs have been described. Major contributions have been made by Edwin Smith Papyrus, discussing the medicinal activity of certain herbal products, mostly in the area of breast cancer. The ancient Unani system refers to the prince and the well-known physician and pharmacist Ibasena, who has given much contributions to the field of pharmacognosy in his book called as Kitab al-Shifa, meaning book of healing. And the ancient Indian system, it refers to the Rig Veda, Atharva Veda during the period 3500 to 1500 BC, and Ayurveda, Ayur meaning life and Veda meaning science. So the life of science. It includes three important documents, namely Charaka Samhita, composed of 500 hymns, which is the oldest document mentioned in the period 2500 to 600 BC. Next, the Shushruta Samhita and Ashtanga Hridaya Samhita, where many medicinal plants or herbs have been discussed, and its potential activity has been also illustrated. So these are some of the pictures which illustrates the significance or the tremendous contributions made in Ayurveda. The ancient China, referring to the Orient herbalism. So this has been mainly given by the Chinese Emperor Shen Nung in the period 2700 BC and Shang in 1766 to 1122 BC, where 365 herbs have been described in a book called as the Native Herbal or First Penti Sao, which illustrates these herbal products into three headings, namely 120 Emperor herbs, 120 Minister herbs, and 125 Servant herbs, which includes high quality food products, mild products, and highly toxic products respectively. The ancient Chinese commentaries on the herbal classic, along with this To Hong Jing's book's findings, have been compiled, and it has been illustrated in the 7 headings composed of 730 herbs, namely stones, grasses and trees, insects and animals, fruits and vegetables, grains, and the last one is named but it is unused. And next we go on to the tremendous contributions by several scientists belonging to the ancient Greece and Rome. To mention a few, Hippocrates, who is called as the father of medicine in the period 460 to 370 BC, Aristotle during the period 384 to 322 BC, Dioscorides in the 1st century AD, and Galen 131 to 200 AD, who have made tremendous contributions in pharmacognosy. So pharmacognosy in modern times relates to the period between 1815 to 1930, and this defines the use of some bioactive constituents, the isolated pure compounds found in plants. To mention a few, scientists like Sidler in 1815 has given a description about these isolated compounds in his book called as Analectica Pharmacognostica, followed by isolation of some pure compounds like narcotin by Duresne in 1803, morphine by Sir Turner in 1806, and emitin by Pellicher in 1817. Followed by this, there has been advancements in the field of science where fluorescence analysis, spectroscopic and chromatographic techniques have arisen in the late 19th century, followed by structural elucidation or characterization of these isolated compounds in the mid-19th to early 21st century. The scope of pharmacognosy is also an important area where we find pharmacognosy has its wide and scope in different fields, namely applied pharmacognosy, where prominent therapeutic activity of bioactive constituents have been described, and this pharmacognosy can also be related to find out the similar type of constituents in related species, and the marine flora and fauna also contributes to highly potent bioactive constituents found in the marine source, and pharmacognosy can also be applied to the genetic engineering and tissue culture biotechnology, where application of secondary metabolites has been employed at a larger level to prepare compounds that is useful for some chronic diseases or unmet diseases. And even pharmacognosy plays a major role in developing some analogs or substitutes in the form of semi-synthetic derivation, where these compounds can be used to increase the potency and decrease the toxicity of drugs. As well, the scope of pharmacognosy still extends its area to cosmetic and perfume industries, and it can also be used as a food supplement, sometimes as a biopesticide, as a dyeing agent, as a pharmaceutical aid in bulk drug preparation, and in herbal drug product industries, where we can go in for the polyherbal combinations for a number of diseases. So to mention a few phytomedicines that are of pharmacognostic significance includes digoxin, which is found in a species called as Digitalis purpurea. This has been used in Europe during 18th century for treating the heart patients. Similarly, quinine and isoquinoline alkaloid found in Cinchona species were long used by the Peruvians and Indians for treating the malarial fever. Similarly, colchicine, which is found in the colchicum species, was used in the treatment of gout by the European peoples in 78 AD. Similarly, there are a number of drugs as listed in the table like emitin, ephedrine, codeine, morphine, atropine, which has significant role and has been used as a potent pharmacognostical aid in treatment of certain diseases. The marine flora and fauna, this is also an important field where pharmacognosy has rooted its significance. There are around 5 lakh species which is reported in the sea and oceans, many of which have pronounced biological activity. Even the Western medicine has established some medicinal products out of it. To mention a few, agar, alginic acid, protamine sulfate, etc. Sometimes the microalgae or the seaweeds are also used as crude drugs obtained from the marine source, which finds use in treatment of iron deficiency as in anemic patients, and also as an additional vitamin given to patients. Apart from this, The marine flora and fauna also contributes to the isolation of some novel compounds like RA, bryostatin, octopimine, etc., which finds use in a number of pharmacological activities like antibacterial, anti-tumor, antiviral, cardiovascular agents, etc. Next is the plant tissue culture, which is found in the present 21st century, which is nothing but an in vitro cultivation of a plant cell or a tissue under a sterile or an aseptic condition, controlled condition, environmental condition, which provides an alternative solution to the arising problems because of the rate of extinction and the decimation of the flora and the ecosystem. So it is nothing but a simple technique which involves 4 steps, wherein a nutrient medium is prepared required for the growth of the particular plant cell or tissue, sterilizing this ex-plant, inoculating and incubating it at predetermined conditions so as to produce phytopharmaceuticals required for the particular disease, and it also been applied to biochemical conversion techniques for cloning as well as for producing some immobilized cells. So why are these natural products important? They contain highly specific stereoenantiomers, they act as sister variants. Always these isolated compounds, because of the constrained ring systems, they provide a challenge to the novel analytical techniques like spectroscopy and chromatography, which ultimately results in the development of new reagents and reactions. To mention a few, herbal medicines of Indian context includes Artemether from Artemisia annua, finding use as an antimalarial, galantamine from Galanthus varroni for Alzheimer's disease, ditisonin from Callistemon citrinus, which prevents the accumulation of toxic byproducts in kidney and liver, and triotropium from Atropa belladonna, which acts as a good bronchodilator. So some important herbal drugs which are of global value includes poppy, saffron, senna, Rauwolfia, papaya, solanum, tulsi, vinca, etc. So the development of pharmacognosy in the modern times started from 1930 to late 1990s, where we find tremendous contributions made, starting with isolation of penicillin in 1928 by William Fleming, followed by isolation of some important alkaloids like reserpin, which has anti-hypertensive and tranquilizing property, vinca alkaloids, namely vincristine and vitblastin, having anti-neoplastic activity, and some steroid hormones like progesterone from diosgenin. So the modern concept involves isolating these phytochemicals and finding out the structural activity relationship, undergoing some partial synthesis of the natural products to provide models for the synthesis of new drugs, and applying the direct therapeutic uses, wherein the biosynthetic pathways have been investigated by some precursor techniques, and also it can be applied to the pharmaceutical aids, and as well in cultivating and collecting some medicinal plants by plant tissue culture techniques, and thereby producing some herbal formulations in herbal industry. So to sum up, the present topic has given a brief introduction about pharmacognosy, its definition, the meaning of crude drug, organized and unorganized drugs, the examples of crude drugs obtained from different sources like plant, animal source and marine organisms, with followed by a brief introduction about the history of pharmacognosy, where the different systems of medicine have been discussed, their origin, contributions made by some scientists, their findings with regard to classification of herbs has been discussed both at modern times as well as in the ancient times. And the next was about the scope and development of pharmacognosy, where we have seen pharmacognosy role in different fields like therapeutic activity, herbal cosmetic industry, in marine flora and fauna. So we have seen how it is used in bulk drug preparation, etc. So the future perspectives, the examples of some phytomedicines which has been used from ancient times has also been discussed. So these modern concepts, or how it can be applied to the present scenario, is also discussed in this presentation. The references for this text, for this topic includes Textbook of Pharmacognosy and Phytochemistry by Biransha and Fundamentals of Pharmacognosy and Phytotherapy by Michael Henrich, with a few findings from the internet. I hope this presentation would be much valuable for you and useful for your notes. Thank you.